Welcome to Standing for Truth. My name is Donny Budinsky, and it is a privilege to serve as your host for today's valuable program. How much genetic change is possible within created kinds? What does modern biology actually reveal about ancestry, variation, adaptation, and the limits of evolution? I am pleased to have PhD biologist Dr. Matthew Cserhati with me again for an interactive program on some of the most important questions in the origins debate as we work to examine the evidence for creation, evolution, common ancestry, created kinds, and also addressing challenges and objections put forth by some of our favorite critics. And so I want to encourage people to bring your questions, bring your objections if you hear anything that we say or present that you disagree with, and join the discussion, because Matthew and I are excited to show you once again why it truly is an amazing time to be a biblical creationist. Dr. Matthew Cserhati, how are you doing today, brother? Welcome to Standing for Truth.
Hello, Donny. Thank you very much for having me on your channel once again. It was great last time, and I'm sure it'll be great this time as well. I'm really glad to be here, and I just want to talk about kinds, about genetics, about evidence for the Bible, all to God's glory. Jesus Christ is our Creator and our Lord and our Savior, so all glory be to Him.
Amen. Praise the Lord. I got to say, the last program that we did together about a month or two ago has received some really good feedback. We are familiar with a lot of the critics and skeptics and their challenges, and I did not see any good rebuttal or counter response from them. So perhaps since we're doing this live and this is interactive, we'll see some objections and challenges that we're happy to engage in our live chat. So format for everybody, in order that we're on the same page, is we will be handing it to Dr. Cserhati to give a presentation. He'll be giving a roughly hour-long, another comprehensive talk. Then we're going to jump into some audience questions, some questions for myself, and just some free-flowing discussion on the topic. So please, if you got a question, you got a challenge, tag me at Standing for Truth or at Donny, and that way I won't miss what you have to say. Okay, Matthew, let's hand it over to you, and I can get your screen up.
Okay, right.
Floor is yours.
Okay, so thank you once again. And so the title of my talk is Kinds or Nested Hierarchies. I'm going to contrast created kinds or Brahmins with the evolutionary tree. And so just to start off again, I've got a channel, Molecular Brahminology. And so if you're interested, you can look at my videos on this channel. I've got several videos on different topics. And so you can also look at my channel as well for other type of content. But just to start today's talk, just to recap, what is Brahminology? What are kinds all about? If maybe you're new to the topic, first let's see how the Bible mentions kinds. About kinds. What is biblical taxonomy? So in Genesis 1, we don't read that. We shouldn't be reading our modern concept of species like Linnaean binomial nomenclature into the text of Genesis. Rather, let's see what it says. Genesis 1 says, "Then God said, let the waters abound with an abundance of living creatures, and let birds fly above the earth across the face of the firmament of the heavens." So God created sea creatures and every living thing that moves with which the waters abounded, according to their kind, and every winged bird according to its kind. And God saw that it was good. So the Bible talks about kinds, and these are larger categories than just like any species. It's a reproductive community, and as you can see that kinds are above the level of species, and so within a kind, different kinds of species can interbreed with one another. So if we, so where does the word Brahminology come from? Two Hebrew words, Bereshit bara Elohim, that's Genesis 1:1 in Hebrew. So bara means he created, that is God created something, and min means kind, and logos or Brahminology, that's the logos in Brahminology, means science or knowledge of something. So Brahmanology is biblical taxonomy, science of created kinds, or discontinuity systematics. And so therefore a Brahmin is a created kind. Using synonyms, it's a type, a group, a class, or a cluster, roughly around the rank of a family. And so this is the approximate level of kinds, although it can be above and below, can be at the level of genus or order, or maybe even class when we're looking at bacteria. What is the boundary of the kind? And that's a very debated question, both inside and outside of Brahmanology, as is also the question of what constitutes a species. So Linnaeus originally held to the fixity of species, and there is a bit of a confusion here that some people think that creation says that species are fixed, they're unchanging. However, species have been observed to interbreed, but their boundaries are fixed. That is, humans, we don't breed with chimpanzees. However, humans have bred in the past with Neanderthals. And so Brahmanology, we allow for natural variation within the boundaries of a kind. At first, Linnaeus set the boundary at the level of the genus. Later, he designated it as the family, so it got a bit wider. And in modern Brahmanology, there have been studies which showed that the superfamily or even order is the boundary of the kind. Superfamily, I did a study in Tyrannosaurids that the Tyrannosaurids superfamily was one kind. The ordo Crocodilia, crocodile kind, is on the level of order. And in bacteria, even the class can be on the level of the kind. The boundary does vary. Let's also remember that we shouldn't put biology into boxes. Whenever we challenge evolutionists, you can't, like last time we showed that evolutionists don't have a clear concept of what a species is. They respond by saying, well, you can't put biology into a box. Well, I'll just use the exact same argument. Where does the evidence lead us? And so I'd like to lay a bit of a groundwork here. Now, if evolution is true, we should see evolutionary tendencies in the fossil record, because fossils are the remains of dead animals. This is a hard evidence of evolution, actually, evolution. So we should be seeing transitions. Also, although we have seen contradictory evidence to the evolutionary picture in the fossil record. We've seen signs of rapid burial. We've seen like ankylosauruses flipping upside down. 70% of ankylosauruses are flipped upside down, because it shows that maybe that perhaps they died in the flood, and so they flipped upside down, since ankylosauruses are top heavy. Or we've seen dinosaurs fighting with one another. We've seen examples of out of place fossils, marine fossils on mountains. I have a set of these marine, I think they're nautiluses, that they, not nautiluses, but maybe spiral shells. And I found them on a hillside in the Gercha Mountains in Hungary. Hungary is landlocked. So why do we find marine animals in a landlocked country like Hungary? Where sudden appearances, bats don't have ancestors in the fossil record. We see groups and not transitions. So overall, the fossil record is leaving evolutionists hanging, and so they turn their attention to genetics. So that's why evolutionists such as Dan Stern Cardinali of Rutgers University, undergrad instructor, he has his own channel, Creation Myths, and he states that correlated hierarchical nature of unconstrained genome regions disproves kinds and proves evolution. And so we're going to examine this statement of his in more detail. He says that constrained genome regions, like coding regions, like genes, they're undergoing so-called purifying selection, which eliminates suboptimal alleles from a population, meaning that coding regions are resistant to change. Of course, that really raises the questions that if the units of selection, purifying selection, are genes, they don't really answer the question, where do the genes themselves come from? I mean, that would really be evolution, but they don't address this question. They just deal with genes as sort of like as if you're shuffling a deck of cards, but they don't tell you where the deck itself arose from. But unconstrained regions, on the other hand, they are free to mutate. And so what Dan is saying is that these unconstrained regions can be used to trace interclade relationships and descent. Although at this point, I do want to question the evolutionist presuppositions by saying that are there really any unconstrained regions? Because this correlates with the junk DNA hypothesis. God doesn't make any mistakes, and so I would say that there really might not be any unconstrained regions. Everything in the genome is used and has some purpose, but we'll talk about this in more detail later. So the Stern-Cardinali test sets up a hypothesis: if kinds really exist, defined at a particular genomic taxonomic level, let's say the family, then these unconstrained genomic regions within the species of a given kind, they correlate well with one another. That is, they have similar gene content, gene order, same type of genomic markers, similar GC%, and that they can share accumulated mutations unique to that kind only. However, if we go beyond the level of the kind, according to his understanding of Genesis, there should be no correlation, no well-defined clades beyond the level of the kind. And so that's his hypothesis. If we go beyond the kind, then this nested hierarchy should stop at the level of the kind and should not continue. Dan contends that this nested hierarchy does continue when we go beyond the level of the kind to engulf all cellular life. However, again, as scientists, we should always ask questions and question authority. So, does a correlation within unconstrained genomic regions above the level of the kind really disprove kinds? Again, because is this what the book of Genesis is really saying? This is our presupposition. Dan's presupposition is that nature made everything. He must find some sort of mechanistic, materialistic explanation for the origin of life. Does a nested hierarchy necessarily correspond to an evolutionary phylogenetic tree? As we'll see later on, that I think that Dan isn't quite understanding the evidence correctly. And of course, are there really any unconstrained genomic regions? If you look at the ENCODE project, whereby they looked at the whole entire human genome, the authors of the ENCODE project said that pretty much the entire sequence space of the human genome is used. There is some sort of function. Let's not stay dogmatic. Let's not say that, well, we just don't find anything. It's all junk DNA. Let's let science have its course. Let's examine these so-called junk DNA segments. Let's ask questions. Let's challenge evolutionary dogmas, and we might even find functions for these alleged junk DNA unconstrained regions. Now, let's look at the. It's rather easy to prove close relationships between species. Let's say a tiger and a lion. There's gene homology. There's similar morphology, hybridization. Tigers and lions, they can hybridize. However, when we go beyond the level of the kind, we do not observe these kinds of relationships. We never see horses breeding with dogs or whales breeding with giraffes. Both creationists and evolutionists, we have the same data. We see the same kind of hybridizations going on. What we see now is that we see kinds. If we build on observable, repeatable, testable scientific evidence, we see kinds. We don't see evolutionary hierarchy. So therefore, our baseline hypothesis is that organisms are really related only within kinds. We see kinds, we don't see anything beyond that. Otherwise, if the evolutionists, such as our friend Dan, if he wants to prove phylogeny, evolutionary common descent, he will have to demonstrate scientifically that these evolutionary relationships exist above the level of the kind, not just inference, not just hypothetically, but if science is equal to observation, testing, and repetition, he has to have direct proof for his evolutionary tree. That, I think, is impossible for him to do, and we'll find out soon. Because what kind of genetic data do we have? Now, evolutionists themselves say that 99% of all species that ever existed are now extinct. Only 1% of species are alive, and of this 1%, 86% of land-dwelling, 91% of oceanic species are still undescribed. There's a lot of taxonomy left to be done, meaning that we only have described about 0.1% of all species, and these are the only the extant species, only the live species. Also, according to Alan Toft, a researcher, DNA cannot survive longer than 100,000 years. So therefore, if evolution is true, if we don't even have the ancient DNA of the 99% of species that are gone, which are essential for building the actual evolutionary tree, because evolution happened to species, organisms in the past. That's what Dan is building his tree on, not the extant species that he wants to make a nested hierarchy of. I think that really Dan is not understanding the proper nature of what he can do with the evidence, with the data. We're not interested in nested hierarchies of extant species. That's irrelevant. So therefore, if we don't have the necessary ancient DNA, which we need to build evolutionary hierarchies, evolutionists do not have the data to even prove common descent. They cannot even get the race going. So imagine that if there's like a race between a creationist and an evolutionist, both of them go to the starting line, but then the evolutionist realizes he is disqualified. He doesn't even have the data to start the analysis. Evolution simply is impossible. They just don't have the data present. So again, this is the tree that I showed last time. We have DNA for the leaves, for these outer branches, but for the deeper levels, this DNA is simply missing. Evolutionists cannot even get the job done. What kind of data do we have of whole genome sequences? We have 23,000 eukaryotes, 22,000 bacteria, 809 archaea, and so these whole genome sequences constitute only about a hundredth of a percent of all species that have ever existed. And so if you're a researcher, if you're data analyst, you know very well that you simply cannot make a hypothesis based on this 100th of a percent of data. You can't make any conclusions. I mean, you need much more data than what we have here. So evolutionism, the evolutionary tree cannot even be proved. And again, if we're talking about nested hierarchies, our friend Dan makes a lot out of alleged nested hierarchies. However, you can basically make a nested hierarchy out of anything. Here's a picture of different sports balls. You can arrange objects into any kind of hierarchy, and you can make like this hierarchical structure out of basically anything. So what Dan actually really needs to do is to prove how evolution did happen, not how it could have happened with any kind of hypothetical nested hierarchy. So his evidence is not hard evidence. He needs to do a lot more to prove his case. And so let's look at evidence against nested hierarchies. Let's look at endogenous retroviruses, viruses, misplaced genes such as Preston, alternative genetic codes, and homoplasy. So let's look at these in order. So now, one thing is that I've been debating Dan on YouTube back and forth, and so I watched his video on created kinds, and so that really piqued my interest. And so I looked at I was watching his video, and he shows this tree, showing what he says shows nested hierarchies, and he's showing that there's like a clade for rodents, for simians. These are endogenous retrovirus DNA sequences that have been aligned in this paper by Benit et al. 1999. Now it's rather interesting because if you look at his tree, below the simians, and I've blown this up, you see that at the very bottom, you'll see that there's an orange question mark, and his tree is cut off. He's not showing the entire tree. And I thought that's irregular. Why is he not showing the entire tree? And so let's actually look at what is going on here. So this orange part here is the missing portion. Again, this article is ERVL elements, family of endogenous retrovirus-like elements, active throughout the evolution of mammals, Lawrence Bennett et al., 1999, American Society for Microbiology. Now, in this orange section, the authors notice that the ERV sequence AS2, which has a star, is for Alota seniculus, the Colombian red howler. It is located outside the simian clade and is mixed in with other mammal species. He also shows 5 dog sequences dispersed among mammals. These are in blue, 2 cow sequences in orange, very far from one another at the top and also at the bottom. There's a horse and a lemur sequence misplaced in this portion of the phylogenetic tree. So we'd like to ask Dan, you did not show this portion of the tree. Would you please comment as to why there's so many mixed up sequences here, especially the one for Alota seniculus? Let's just also make a point here that this tree was made based on 87 ERV sequences from a 360 base pair region of the Paul gene. It's a conserved gene region. That's the POL gene. So it is, it codes for something. It is not unconstrained. It is a coding region. So I think that maybe that Dan even misunderstood his own the paper that he was showing to his audience. So his presentation of the data was actually quite questionable. There are contradictions in his presentation which really refute his conclusion of nested hierarchies, showing what he thinks is common descent. The authors say that one interesting exception among the simian sequences is the New World monkey AS2 sequence, the black triangle, with a blue star, which falls outside the simian group, might correspond, as proposed for some null sequences, to an ancestral sequence not subjected to the simian burst. So what he's talking about is that there's these ERV sequences in the simian, in the monkey clade. There's like a burst of sequence change. And of course, another question is, why do these sequences suddenly undergo these bursts, these rapid mutational accumulations? Authors don't comment on why there was they don't offer any kind of explanation or mechanism as to why this particular sequence was not subjected to this simian DNA sequence burst. So it seems that it's just there. They have no explanation as to why this monkey sequence is mixing in with these mammal sequences. And since we're talking about endogenous retroviruses, let's also talk about a bit of a debate or back and forth we're having with Dan Stern-Cardinale about viruses. Let me see. I don't think this is what I want to show, but okay. Sorry about that. Okay, yeah, I am on track. Okay, I'm going way too far ahead. Okay, I got a bit too far ahead. Okay, so let's talk about viruses. And so I wrote a paper in the Creation Research Society Quarterly about viruses. Do viruses form Brahmins? And so I used an algorithm that I developed called the gene content method. which basically compares to two species and compares their similar gene content. Based on this, based on gene content similarity, I can deduce whether they belong to the same kind or not. And so my conclusion was that viruses, they act a bit differently compared to cellular life. And Dan actually jumped on my video, and so he his argument basically was that, and again, so here's the gene content method. On the left side, you can see that in this case, if you have two species with similar number of genes, you have high similar gene content. These two animals or these two species would be from the same kind. On the right-hand side, we have two species with different number of genes, low similar gene content. This would correspond to different kinds. And so the overlap divided by the union will tell us how similar two species are based on their gene content. And so what I found is there are 8 larger well-defined virus clusters. And so each cluster of viruses, they either infect one genus of prokaryotes, or like bacteria, or viruses can also infect multiple genera. So I found that viruses, they do form clusters, but again, do they really behave like cellular life? I found that there are kind of differences. And so Dan is saying that since that I used my method, I found that viruses they form kinds. And so interestingly enough, his argument is that viruses form kinds, therefore cellular life, which is different, must follow common descent. I don't think that's logical. I don't think that's logical at all to say. And so he's saying that I used my gene content method to demonstrate that viruses have independent origins. And so he's saying that therefore cellular life forms have common ancestry. That is much too much of a leap to make, because he's basing his conclusion on nested hierarchies in unconstrained regions. I did not analyze that. Dan is saying that his H-null hypothesis is that viruses form kinds, therefore cellular life does not. This is a big non sequitur. It simply doesn't follow. Dan's logic is flawed. Furthermore, it's interesting to say that viruses have an independent origin, since their genome structure is fundamentally different from one another. And it's very interesting to note that this is a technique that is used in baraminology to separate created kinds from one another. So in essence, Dan himself is thinking very much like a baraminologist. The other major point that I think I have to make is that Dan is inconsistent. First of all, that he's comparing apples and oranges, viruses with cellular life. I don't think that Dan's research methodology holds up, because in research, and I think that Dan is mainly like a lecturer. I don't know. I didn't see that he has published papers in PubMed, whether he's done any actual research. In research, I mean, I have done research, bioinformatics research, that if you apply one algorithm to one data set, you have to apply the exact same algorithm to another data set. And so that is exactly what I've done. I applied the same algorithm to primates. This is the results of the gene content method. And as you can see, that this was published in CRSQ, and using the same algorithm on the same data, I found that, for example, humans, Neanderthals, Denisovan, and modern humans, Homo sapiens, in the bottom left corner, they form a group, they form kinds. And so we see examples of other kinds. We see great apes, Pongo, New World monkeys, Cercopithecines, and several other groups forming kinds. And so if we follow Dan's logic, we can say that this gene content method finds kinds in viruses and also in primates. I'd also like to add that my method, the gene content method, was actually pre-discovered by a group of evolutionists in 2009, a Dutch group, and they actually discovered this gene content method before I did. They published a paper on it, and so Dan is sort of like saying that, well, Matthew, based on your logic, you found kinds, and so therefore you fit your own argument. But no, I mean, I used my gene content method on both viruses and also cellular life, and I found kinds in both cases. I mean, if I find kinds in viruses, I use the same algorithm in cellular life forms, in primates. It should stand that I'm finding kinds in primates, and so I just think that Dan is using bad logic and bad research methodology. Maybe he should take a refresher in both areas. So anyway, if we keep going, we can also look at misplaced genes. And so there's a paper by Lee et al. From 2010 in Current Biology. They took a look at the pressin gene, which plays a role in echolocation interestingly in bats and whales. These are both groups which use echolocation. And so at the very right-hand portion in green, we see this clade of microbats, which are echolocating. And we also see a group of megabats in blue. And what's interesting is that we see in red the bottlenose dolphin, which clusters closer to microbats, which are echolocators. And so, what does this mean? Does it mean that the dolphins and microbats are evolutionary relatives? As you can see on the image on the right, that we have like a bat dolphin. This would be the conclusion that we can draw from this evolutionary paper, that if the research is true, then indeed, based on the pressin gene, that bats are related to dolphins. Of course, no evolution would take this seriously. This is not what evolutionary trees are based on. And so this is one big contradiction of self-contradiction in evolutionary trees grouping bats with dolphins. The authors themselves saw this, and it really piqued their curiosity as to why dolphins are clustering with microbats. And so they came up with several scenarios. Is it horizontal gene transfer between sea and flying animals? Rather unlikely, because one is in the sea and one is in the air. How would gene transfer occur between bats and dolphins? It's very unlikely. They also excluded DNA contamination. What about duplication and differential gene loss? According to these authors, they see that each mammalian genome only has one pressin gene, so it's highly unlikely that they have that if there was duplication and differentiation, that all these species in unison gained and then lost the same number of genes in unison with one another. They also asked, is it general long branch attraction? They also excluded this because they found that the branch leading to the dolphin is shorter than rabbit, which they consider not to be misplaced. We see that at the bottom, the fourth to the bottom is rabbit, and that branch is longer than the branch leading to bottlenose dolphin. They don't have any problems with rabbit, and so this branch is longer than the branch leading to the dolphin. They just can't really find any explanation as to how or why dolphins are clustering with microbats. The explanation I would venture is that this pressin gene is a created functional unit, and so dolphins and microbats, they both echolocate, and so it's small wonder that you find this functional unit, this DNA sequence in both groups of organisms, in the dolphins and also in the bats. Let's look at alternative genetic codes. And so from high school biology, you might remember that the genetic code translates RNA to proteins. That is RNA triplets to amino acids. That's a 3-to-1 correspondence. And so there's like a very general genetic code which maps each of the 64 base triplets. You know that if you have one codon is a base triplet, you have 3 positions in which you can place a possibility of four bases. And so there are 4 times 4 times 4, that is 64 different triplets, DNA triplets. And so there are 20 amino acids. And so this is how you can map codons to amino acids. There's one very widely used genetic code, but there are about 3 dozen alternative genetic codes which exist all across life, from bacteria to fungi to echinoderms. These alternative genetic codes, they do occur in various places. They can either exist within individual genre, but also in larger clades. They're found in the nucleus, but also mitochondria also have their own alternative genetic codes. It's interesting because evolutionists think that many genes have been transported from the mitochondria to the nucleus, and so if the mitochondria have their own special code, how can they overcome the differences in this genetic code between the nucleus and the mitochondria within the same cell, within the same species? That's a big hurdle for their theory to overcome. Also, it's interesting that genetic codes, these alternative genetic codes, they show homoplasy, which means that there are specific alterations in the universal code which tend to be preferred compared to other alterations. It's like you're making the same change in several widely different groups which are positioned on very different parts of the evolutionary tree. And so the question is why is this happening? And so, for example, if we have 2 related species which are similar to one another in the same clade, if we have alternative genetic codes, a portion of one species' protein sequences can either be modified, can be mistranslated, it can be extended if perhaps a stop codon is switched to an amino acid, or if it's truncated, if an amino acid is turned into a stop codon. And so all sorts of different things can happen. If you change one codon, it can possibly mess up a lot of your proteins. And so this is a big hurdle for evolutionists to leap. It really introduces a lot of discontinuities between related species. And again, there are different clades of organisms which have specific alternative genetic codes. And if we think the way Dan Stern-Cardinale does in a Bermanological sense, let's follow his Bermanological way of thinking. So if viruses have independent origins based on very different genome architecture, then we can apply alternative genetic codes which bring about very different, they're sequentially very similar, but the resulting protein is very different. So we can apply Dan Stern-Cardinale's Bermanology thinking to alternative genetic codes, and we can use these as markers to delineate different kinds. So thank you, Dan. Here's some examples of alternative genetic codes. The UAA and UAG stop codons have allegedly been switched to glutamine, that is CA and CAG, at least 5 times in eukaryotes. Big question is why the exact same stop codons 5 separate times in eukaryotes. In these groups, ciliate, spinalalgae, diplomonads, because if you think without a stop codon, the UAA and UAG, they are stop codons. If these 2 stop codons, if they switch, then the protein will become elongated. And so that is not good for evolutionary theory, that with this change in the genetic code, you'll suddenly have very different proteins within related species. So again, I just wanted to show, it seems that the PowerPoint wasn't taking this, but the probability for this happening is that the stop codon transforms into I think it's a glutamine codon, and so the chance for this to happen 5 times independently is like something like 10 to the minus 8th. Unfortunately, this got cut off, so I'm just trying to explain the probability for this. Okay, so we also have functioning non-coding regions. Again, the evolutionist, my friend Dan, he relies on nested hierarchies in unconstrained regions. That is where the sequences are free to mutate. The question is how much unconstrained non-coding regions are there, because the ENCODE project came out that virtually the entire human genome is used. And again, why should we just stop? Why should we just dogmatically say that everything is junk? It's similar to saying that we have 100 vestigial organs in the human body. They don't have any use, or just evolutionary throwbacks. But with these vestigial organs, we found that with some research, we have been able to assign functions to these vestigial organs. Like we found that the appendix has a role in the immune system, and so we look at these non-coding regions. I mean, why can't we use our scientific fantasy or intuition? Let's just do analysis. Let's do science, and we'll overturn these religious evolutionary dogmas which say that these non-coding regions are just junk. They don't have any function. So junk DNA hypothesis has been discredited for decades. I don't understand why Dan is holding onto this so religiously. And again, we just have to do neuroscientific studies, and we do. Scientists are finding functions for example, for pseudogenes. I've just recently had a review by 2 Russian authors that a lot of pseudogenes they are expressed. They take part in, for example, in mRNA decoying to prolongate the lifespan of the mRNA in the cytosol. Many of these pseudogenes are implicated in cancer, meaning that even though they undergo mutations, their function is being degraded. The degradation of their function causes things such as cancer. So don't just write off pseudogenes as junk DNA. They may, for example, they take part in regulation of gene sequences. Some evolutionists, Bakel et al. And PLOS in 2010, they allege that the genome is not pervasively transcribed. However, in their paper, they used the program repeat masker. They masked out regions with at least 20% repetitive content. So again, there's another assumption that repetitive DNA sequences are useless. How can you say that? I mean, like telomeres are repetitive, but they have a function. Telomeres are these sequences at the very caps of chromosomes. They have a protective function for the chromosomes. However, the Repeat Masker website itself says that up to 50% of the human genome is repetitive in nature. And as improvements are made in detection methods, this number is expected to increase. Well, I mean, if you think about it, if 50% of the human genome is repetitive, that's a lot of excess baggage to carry on between cell divisions. And so if evolution is true, I think that evolutionary processes would really streamline these repetitive sequences and get rid of all this junk, but yet it's still there. We can still entertain the hypothesis that these repetitive sequences, this alleged junk, has some sort of function in the human genome. So let's do science. Let's see if there really is a function for these repetitive sequences. Another thing I want to address is it's another debate. We critiqued Dan Stern Cardinali and Gussie Gibbon, Erica. I'd like to talk about some results in the debate between my friend Dr. Jerry Bergman and Dr. Chris Thompson. They had a debate, and they looked at mouse kinds and also dog and fox kinds. And so Chris Thompson was challenging Jerry on where he puts the level of the kind in mice. And so he referred to this paper by Stepan Schenk in PLOS 2017. This larger group called Murida contains 1,620 species, which are 30% of all mammals, with 6 families of muroids. These are Cricketidae, Muridae, Nesomidae, Calomycidae, Spalacidae, and Platacanthomidae. And so, the question is, where do we draw the line for the kind? And so I examined the paper that Thompson was referencing. During a debate, you don't have the luxury of taking the paper that your opponent is talking about and examining it in closer detail. So let's look at these assumptions. Let's look at what's going on. So Thompson equates families with kinds. So he's equating the 6 muroid families as each as one kind. Although, in Brahminology, that's sort of like an approximate level. Just as evolutionists, they also have approximate levels for what counts as a genus or a subgenus or a species. And so doing my research, I found that the order Crocodilia is one kind. These are the crocodiles, the caimans, alligators, and the gharials. These are 4 groups. And also using extinct species, I was able to unite existing and fossil species into one kind, the order Crocodilia. Another recent paper of mine showed that based on mitochondrial sequence similarity for mammals, the boundary in mammals can be between the genus and the family. So it is true that the boundary of the kind can vary from the level of the genus to the order in vertebrates, just as for evolutionists, the boundary of the genus or the species also varies, and they also don't have a limit for what the species is. Because again, let's not try to put biology into boxes. And so if you look at the paper, these 2 authors, they mentioned that during the evolution of muroids, the speciation rates accelerated in either 4 shifts, or in 20 shifts, or in 24 shifts, according to various authors. And so 20 to 24 shifts, that might be like acceptable, that, and these are accelerated speciation rates. I wonder why does speciation accelerate in evolution? Because that would imply a whole bunch of mutations happening at once to jump from one clade to another. I don't think that evolution really should be allowing accelerated mutations. That sort of speaks against evolution. That speaks for disjunct clades that are discontinuous from one another. That's a discontinuity principle in Brahminology, as Dan Stern-Cardinali discovered for himself. Anyway, so what causes these speciation shifts? Why are their numbers so different? Why 4 according to one author? Why 20 according to another author? I think that this again shows that evolutionists don't have a clear idea of what speciation is. Could these shifts maybe be adaptive radiations? That's a possible explanation. Creationists, we do have an explanation. It's the age hypothesis, meaning that when the flood came to an end, in the post-flood world, there were all these new ecological niches. There were all these areas that animals could come into and populate. It was like a brand new pristine world. And so could it be that when the animals came off the ark, they had a lot of new niches that they could adapt to? And so that's what prompted many different groups to rapidly adapt to a lot of new niches. It's like, imagine that you come upon an empty city, and there are these apartment complexes, there are these homes, these garden homes, there are these high rises, and so people, these new niches, and so people populate these different areas very rapidly, and so there are these new living spaces, and so these animals, they adapt very rapidly to these living spaces. When we look at the evolutionary tree in Stephen and Schenk's paper, they only analyze 75% of muroid genre 250 or 330. Their tree is also based only on parts of 6 genes, 5 nuclear genes and 1 mitochondrial gene. They also say that gene sequences were removed if concordant topologically with evidence from other genes. So gene sequences were removed if they were concordant topologically with evidence from other genes. That is, largely different sequences were excluded. So this is cherry picking the data. If it doesn't fit their evolutionary presupposition, then they just remove that data. They also rate that the accessions with unexpected placement from analyses, if their discordance was great enough, meaning that if they found sequences which were very discordant, they were also removed. So again, this is homoplasy. They just remove data which doesn't fit like a generally overarching evolutionary hypothesis. And this is really just cherry picking. Again, it shows that your ideology even influences your data collection. And so the way they collect even data which is supposed to be neutral is not doesn't escape selection based on their evolutionary ideology. They also rate that a best fit model was chosen by selecting one of 57 DNA substitution models that fit the data best, while avoiding over-parameterization with the Akaike information criteria, meaning that they generated 57 different tree models, and they picked the one that fit the model the best. And again, obviously these authors did a lot of work, but this type of research methodology is unacceptable because you're selecting a result which reinforces your initial evolutionary presupposition. They simply remove sequences that mess things up, like the Preston gene in bats and also in dolphins. And so basically, if you put evolution in, evolution will come out. So I don't think that we can really give Thompson any points in his debate with Dr. Gerald Berman. I mean, really putting evolution in, and of course, they're going to get evolution coming out the other end. I mean, it's biased research, but of course, I mean, evolutionists can get away with this because evolution today is the ruling paradigm, and so they can simply allow this for themselves, whereas they critique creationists. So again, let's also look at another point that was raised in the Bergman-Thompson debate. It's the dog and or fox kind. And so there was a paper in the Journal of Creation in 2019, and they show that there are 400 different breeds of dogs. And we see that dogs can be very different from one another. And so the question is, do dogs, apparently they come from the wolf. And so, what about foxes? Are they part of the canid kind? What's the situation with them? Because you see that Thompson was challenging Bergman that we have these 3 species. They look very interesting. We have the raccoon dog. We have the bush dog. The bush dog in the middle, and the maned wolf on the right. And so they're very interesting looking species. Are they dogs? Or are they foxes? Or are they something else? Or are they maybe raccoons? The one on the left sort of looks like a raccoon. And so what's the deal with these species? Again, but looks may deceive. There may be great morphological variation within a kind. However, even within the species due to sexual dimorphism. I mean, there are humans who are 2 feet tall. There are also humans who are 8 feet tall. You might confuse them if you only look at their fossils. These humans you might consider them to be from different genre. And so this is a morphological evidence. And so that's why I like to study molecular Brahmanology, because in my view, now morphological studies are very interesting, but you can also buttress them with evidence from molecular data like mitochondrial DNA or whole genome sequence analysis. And so let's look at what the DNA sequences have to say. So I took mitochondrial genomes from about a dozen or so, maybe 10 species of dogs from Canidae, and also Oh. Oh, unfortunately, I think I might have put in the wrong tree. Oh, yes, unfortunately. Okay, I'm showing the wrong data. Basically, I found that based on, let's not look at the left-hand side. I made a mistake. I was analyzing canids and also vorpids, foxes and dogs, and I found that based on the elbow plot on the left, on the right, that the elbow plot is a good tool to find different groups of different kinds. And so I found that if you look at there's an optimal number of three kinds in this data. Where there's a big break in this elbow plot, the elbow plot shows the total within sum of sequences per the number of clusters. And so we found that there's an optimal number of 3 clusters. And again, the heat map is messed up. I apologize for that. But we basically, it came out that there is a cat kind, a dog kind, and also a fox kind. The Hopkins statistic also shows whether there are real clusters in your data. And so the higher this statistic is, shows that there are real kinds in your data. And so 0.8 means that there are decent clusters in your data. So I'm really sorry for the technical problem, but basically the conclusion that I was able to draw is that that the dog kind and the fox kind are two separate kinds. Now, getting to the end of our presentation, we're talking about kinds and species and evolution and variation within a kind. And so we're talking about what is the border of a kind, what is a species. And so where are we going with all this? Now, evolutionists themselves, they also reject typology or essentialism, which is basically the study of created kinds. Basically, the typology or essentialism says that there are a group of organisms which stay essentially the same all throughout time, and so they vary within the same kind. So evolutionists are rejecting typology and essentialism, but what do they put in its place? Again, many evolutionists do not have a clear concept as to what a species is. Pence, in his article on A History of the Idea, in Evolution, Education, and Outreach, he says that the dividing line between a species and a variety is an arbitrary one, drawn for the benefit of the biologist. So he's saying that basically that a species is a logical construct. It's just it's not a real biological thing, and so because all life forms they're evolving through time, and so species change all the time. You can't really define what a species is. But then let's take the same type of thinking and let's apply it to humankind. And so I think it's interesting that humans, we very instinctively talk about the humankind. We really recognize kinds in nature. Now, who would we exclude and who do we include as humans if the boundary of the species is as fluid as Pence is making it out to be? Why are we saying that the boundary of species for the human species can shift? Are humans the third chimpanzee species? What if we go the other way around? And what if we exclude people from humanity? Different, and I don't want to go there because this is exactly the way that for example, the Nazis thought about during the Second World War, and led to the extermination of various people groups, gypsies, and Jewish people. And so, if you think this way, that there is no real human species, this can go really sideways very quickly, and it's just a bad idea, bad thing to think about. Rather, species are real things. We can readily recognize species through disjunctions. Species and kinds are real things, and so really the evidence really supports kinds as opposed to evolutionary hierarchies. So in conclusion, we can say that the fossil evidence is contradictory. We don't have ancient DNA, which would be the actual evidence that Dan Sternquist and I would need to prove molecular evolution. If we did have this ancient DNA, we could also prove that organisms are young, because DNA breaks down very rapidly. If we do have this ancient DNA, it would prove that organisms are young. We can also examine the phylogenetic relationships of ever more species. I'd love to do this kind of study. Evolutionists, since they don't have this data, cannot even get started proving molecular evolution. They have to exit the race before they even turn their engines on. Therefore, the evidence this way points unequivocally to kinds. Nested hierarchies in unconstrained regions, if there really are unconstrained regions, don't logically prove evolution. And if with scientific research we can find functions in these alleged unconstrained regions, homoplasies are ubiquitous and create contradictory trees. Homoplasies are defined as they are similarities which occur in very disparate, very distant parts of the evolutionary tree. For example, mollusks like octopuses and squids, they have chambered eyes with retina, and also with a lens which is very similar to human eyes. They also found that jellyfish also have things called rhopalia. They have eye-like structures on the very edge of their bells. There's some jellyfish which have these so-called rhopalia. These rhopalia, their eye-like structures. They also have this lens-like structure which is used to focus light. So eye-like structures in jellyfish, in octopuses, and humans. And so that is really something difficult for evolutionists to explain. Homoplasies are like the bane of evolution, and actually quite a large percentage of different morphology characters are homoplastic in nature. Homoplastic characters, they're like those types of traits, which if you put into an evolutionary tree, it makes certain species, it just really upends the evolutionary tree. For example, we saw an example in the Preston gene, which displaces the bottlenose dolphin and makes it a sister species to microbats. Also, we've seen that alternative genetic codes, they mess up evolutionary trees, and so we've seen that fossils don't work. Non-coding regions are finding they're finding a lot of functions for non-coding regions. Homoplasies mess up evolutionary trees. The alternative genetic codes are also a problem, and so evolutionists, they don't have the ancient DNA. They don't even have the stuff to even make a molecular genetic-based evolutionary tree, and so for people like Dan Stern and Kodinai and colleagues, what's left, I would say that basically I have to give it to them that they do have a lot of chutzpah for trying to defend what I think is pretty indefensible. So we've seen a lot of evidence here which really contradicts evolution, and so with that, I think I'm done with my presentation. It's a little bit over an hour, and so I think that now we can start with questions.
Dr. Cserhati, excellent job. It's like you've done this before. You hit exactly the hour mark, and that was comprehensive. That was thorough. Yeah. Before I even get to my first question, I have to say that you did an excellent job there. I appreciate all the time and work and energy that you put into that very thorough presentation. I would not want to be an evolutionist, somebody who believes I'm related to chimps, monkeys, banana plants, and grass, trying to refute that because that was top notch. And answering the question of ancestry is important because that's the major question: is all life related through common ancestry, or is separate ancestry true? That humans are special and unique, created separate from any other form of life. That's what the Bible says. God
Yeah.
created people, Adam and Eve. From the beginning of creation, he made them male and female. And so they're unique and separate from chimps and grass. So I'll start with this because I have a lot of questions based on your presentation. You tackled at least a couple critics and their challenges, like Dr. Dan Stern Cardinal, and then also Dr. Chris Thompson engaged some of his points from his debate with Dr. Jerry Bergman. Perhaps we'll start with the topic of unconstrained sequences, junk DNA, pseudogenes, and ERVs. They're kind of all in one in a way.
Yeah.
Because, and I have several papers in my PowerPoint presentation on ERVs where they will say directly in the paper, what was once thought to be junk DNA is now known to be functional in helping to determine cell types in the immune system, embryological development, so on and so forth. So point is, the evolutionary community themselves are admitting that they've assumed DNA units like ERVs and other classes of retrotransposons were non-functional junk. Now we know they're functional. The pseudogenes, you went over several functions for what are assumed to be pseudogenes. They can help regulate their protein coding counterparts. They can act as a decoy. Actually, perhaps could you reiterate a couple of the functional roles associated with pseudogenes, because that is a huge argument that's still brought up, is the argument from pseudogenes.
Okay, so for example, pseudogenes, let's say you have a gene and it produces a messenger RNA which goes into the cytosol. Now the messenger RNA, once a protein is processed, once it's formed, the messenger RNA is not going to stay there forever. It's going to be degraded by proteins or enzymes. Now, what the pseudogene does is it is transcribed, and now its sequence is different from its parental gene, but it's still similar enough so that it can sort of act as a decoy for these degrading enzymes. So it's sort of like they want to keep the parental mRNA in the cytosol, and they're sort of like decoying these enzymes which would otherwise break down the messenger RNA. And so otherwise, pseudogenes, they also take part in gene regulation. They have quite a lot of other functions. I mean, these two Russian authors, they wrote a whole review paper describing the different functions for pseudogenes. Simply the fact that pseudogenes take part in cancer when the cancer cell starts, it undergoes dysbiosis. It would mean that these pseudogenes, they do have some sort of function like in the normal cell cycle. And so when the cell becomes cancerous, then these pseudogenes, they start, breaking down. And so their role, their function in the cell cycle also goes awry. But I would think that pseudogenes, simply the fact that there are pseudogenes, I mean, first of all, evolutionists, they're not explaining how do their parental genes come about in the first place. Evolutionists almost, I mean, they never do this. Like to explain how random genetic sequences, how they give rise to gene sequences, they just don't give any explanation. As to the pseudogenes, they're also degraded sequences, and so they're going in the opposite direction. They're not building up genetic information; they're degrading existing genetic information. That's devolution at best. It's not evolution. You need to go the other way if you want to explain evolutionary theory. And so I would also add that the degradation of genes into pseudogenes, I think, is proof for the fall into sin. When I mean, originally God created everything good, but Adam sinned, and so now there's death, disease, entropy, increase of entropy. Things fall apart. rather than being built up. And so I think that pseudogenes are really prime evidence against evolution.
That's a very helpful response. So, as creationists, would we then have to argue that, okay, because of the fall and since the flood 4,500 years ago, we know mutations accumulate rapidly in autosomal nuclear DNA. Last time I checked, roughly 100 new mutations per person per generation. Most of those damaging, harmful, disease-causing, slightly deleterious. So we would expect some broken genes. So some, I guess, what you could call pseudogenes, but also recognizing that a lot of what are called pseudogenes may actually just represent created DNA units. Matthew, how would we parse that out as a creationist?
I think it could be both. Both scenarios, they both fit into the creation model. And so, if you think in the evolutionary model, I mean, they say that, well, I mean, it's genetic change, and change is evolution, because evolution is change, but it's change going in the wrong direction. So it is evolutionists could use this for their model, but it's not sufficient evidence. It's not really really groundbreaking evidence for the evolutionary model. It fits much nicer into the creation model.
So if we find pseudogenes that are just original created DNA units or genes that are functional in several of the functions that you mentioned, I find the decoy mechanism very interesting.
Yeah.
And perhaps you can correct me if I'm wrong. That pseudogene, in order to even perform that role, has to share sequence similarity with the so-called parental
gene. Yeah.
gene,
Yes.
Which is incredible, because then evolutionists will say, well, why does it's also analogous to the argument they use for ERVs. Why do ERVs share sequence similarity with retroviruses if they didn't come from retroviruses? I like to point out, well, they require that sequence similarity to carry out many of their roles, like fighting against retroviruses, acting in viral mimicry, so on and so forth. So is it the same thing with the pseudogenes, that essentially many of these pseudogenes have to look like the parental genes in order to carry out that decoy?
Here's what I think. I think that the word pseudogene has evolutionary baggage, because the term that pseudogene, it's suggesting that it's a degraded version of a gene. And I mean, if you think about it, originally during the history of genetics, for example, Beetle, I forget his first name, but originally they came out with the hypothesis that it's one gene and one protein, and it's like a one-on-one correspondence. But then they did more research, they studied the genome, and so they found that there are things like multiple alternative splicing, that you can get multiple different protein variants from the same gene. Or like, what is the definition of a gene? That they thought that first it's only like coding sequences, then it has a promoter, it has a terminator region, like a poly A tail. However, they as more research was done, researchers found that there are enhancer elements which can even be found on other chromosomes, which enhance and induce the expression of genes on a totally different chromosome. So, and I think this is very difficult for evolutionists to explain, that you have to have concerted changes on multiple chromosomes all playing together at once. And so we see that as we know more and more about the genome, that it's not just junk DNA. It's not like almost all empty genome. That as we look at more and more DNA sequences, we find more and more functions for them. And as Robert Carter from CMI likes to point out, that the genome is 4-dimensional. It's really high dimensional. It's so integrated at so many different levels, so many different genetic elements, transcription factor binding sites, alternative splicing, enhancer elements, insulator elements. There's so many things which all have to be exactly right to induce the expression of a gene so that it plays its role properly. And so genes and genomes are such an exquisitely, they are a really delicate construction, so that one change or one mutation somewhere can easily induce like a harmful mutation in the genome. That's why researchers are discovering more and more harmful genetic mutations. There's a special database, the Online Mendine Inheritance in Man database. And so we see that mutations, they don't build up genetic information. They don't build up the genome. Rather, mutations, they destroy existing genetic information,
Right.
which evolutionists they can't explain how the genome got there. They can try to explain how genes are shuffled left and right via horizontal gene transfer, or exon shuffling, or transposing genes from one part of the genome to the other. But as to explaining how new genes arise via random mutations, they don't have any good explanation for that.
Well, that's a great extensive response. I appreciate it. I do like the way Dr. Carter puts it, that the genome is 4-dimensional, chock full of meta information. It has multiple overlapping codes. I mean, how do you evolve a system like this? And so going back to the pseudogenes, I think you made a very important point. The so-called pseudogene did not call itself pseudogene. An ERV did not call itself an ERV. The evolutionists got to it first, and there's evolutionary baggage there. It might be best to say this is a created unit of DNA function, because it's the evolutionary community that assumes all DNA differences are either evolutionary leftovers, viral fossils, the result of mutations. But as you've pointed out, the vast majority of mutations are deleterious. They're not going to build information. So my question is this then: when an evolutionist challenges us and says, and we often see this in presentations, I just did a formal debate recently on genetics, and one of the evolutionists that we debated, was a 2 versus 2. He had a phylogenetic tree, and he showed pseudogenes, or so-called pseudogenes, genetic mistakes shared between humans, chimps, the lesser apes, monkeys, going down the line with primates, showing this nested hierarchy. So I guess I'll say this as the question. So their challenge is, well, why do these genetic mistakes follow the hierarchical pattern? Why do we share them with chimpanzees? But you're saying, and feel free to speak to this, that a lot of those might not actually be pseudogenes. They're created units of DNA function. Therefore, we would expect to share some with the chimpanzee and other creatures.
I mean, like pseudogenes, you can look at them as like molecular appendixes. I mean, we found a function for an appendix. I mean, we just have to do scientific research. We have to use our scientific imagination. We might find functions for these alleged pseudogenes. You talk about like ERVs. Anything that Liu and Soper, they talk about ERVs in their paper. What they point out is that there are 2 separate lineages of ERVs in, I think, in New World monkeys and also in Old World monkeys. And so what they're saying is that in 2 separate evolutionary lineages, we find that these ERVs, they arose separately. They have 2 separate origins. Again, this is proof of homoplasy, and not which goes against evolutionary theory. So I think that in this case, when you look at the ERV sequences themselves, they're structurally different enough that you're forced to say that these are 2 separate lineages of ERV sequences.
Well, that's an interesting point.
Separate, independent origins.
Independent origins.
Which is homoplasy.
So, Cserhati, we can. Does this go back to what we were discussing in our first program? It's the differences that make all the difference. Cserhati can account for homology and homoplasies, because we can account for extreme differences because we believe in independent origins, where the evolutionary community, they struggle with those differences, whereas those differences actually help us to draw lines in kinds.
Yes. So like homoplasy, and again, evolutionists don't like homoplasies. And I have a copy with me of the only single book that has been written on this subject by evolutionists. They've only written one book on this subject. They sort of don't want to look at it because it contradicts their theories. So again, homoplasies are good for creationists because how can an evolutionist explain a homoplasy? I showed the example of like the same stop codon to glutamine transition, and it happened 5 separate times. Now, for that to happen, that has like a probability of 10 to the minus 8th. Has a
Wow.
very low probability. And like, why does the same change happen? I mean, it's like winning the lottery 5 separate times.
Hmm.
And so, why do eyes evolve in jellyfish, in octopuses, and in humans? Like a very similar organ for sight. Why does it happen in these 3 very different portions of the evolutionary tree? And so it's hard for evolutionists to explain, but recreationists, we can see that these are design functional units that God used to build the body plans of various organisms. He created wings for bats, birds, and insects, eyes for octopuses, humans, and jellyfish.
Well, again, that's a great response. And so, focusing on this constrained versus unconstrained sequences argument that you did a fantastic job there countering and addressing, I want to engage this part about mutation accumulation versus designed alleles. As creationists, we need to separate the two for sure. And then genome-wide functionality. You discussed ENCODE, how they revealed that over 80% of the genome is biochemically active.
Or even like 100%.
That's incredible. So to suggest, and we just did a show on genetic entropy last night, and we pointed out that even with a lot of junk DNA, the mutation rate is still too high for evolution. But now that we know that the majority of the genome is functional, that means evolution's impossible. That precludes the mutation rate's too fast, selection's too limited, they build up, they degenerate.
And also, I mean, one of my big beef about this junk DNA hypothesis is that you can observe like a symbiotic bacteria, and these symbiotic bacteria, they're very interesting that they colonize either, let's say, the intestines of insects, or they colonize like the wall, like the different cells in plant roots. And they see that as they colonize these different living spaces, their genome gets reduced. They get rid of those genes which they don't need because, let's say, if the host provides, let's say, sucrose, then these bacteria will no longer need enzymes to produce this particular sugar. And so therefore, to keep this particular gene always handy in their genome is unnecessary, because during cell division, remember, you have to copy the whole entire genome sequence, every single last chromosome, every single last gene. And so why keep on copying something which is like a dead weight? You know, it's like, if you're moving to a new city, why do you keep dragging on old papers from 15 years ago? It's best if you just throw it out. So I mean, it costs a lot of energy and a lot of nucleotides to keep on maintaining those dead genes that you don't need anymore, which which the host is supplying for you. And so junk DNA, I just think, is a very, very, very bad hypothesis. It's a junk hypothesis.
Well, that's why the rescue device, which you just dismantled there, that I hear from people like Dr. Dan Stern, Cardinal, and others, they'll say, well, that 80 to 100% activity, biochemical activity, is just spurious or noise. But I think you just nailed it. Well, wait a minute. Transcription is an energy-intensive and resource-intensive process.
Exactly.
Every time you add a single nucleotide to a growing RNA chain, you're consuming what? Energy.
ATP, nucleotides, sugars, all sorts of stuff. I mean,
so you're telling me that evolutionary processes are just going to keep all of this excess genetic baggage rather than over time weed it out to preserve energy for the cell?
Yeah, well, yeah, and especially when what we observe in lots of these bacterial species, that genomic streamlining does occur. It does occur. It's a real phenomenon. I mean, I think this junk DNA hypothesis is really just made up and it's being disproved, but some people really are just holding to it dogmatically.
So when evolutionists challenge us and say, well, why do we find homology and nested hierarchical patterns in junk DNA sequences or sequences that don't have a function? What you're pointing out is there's a built-in assumption in that challenge that those sequences and genetic markers are non-functional to begin with.
Yeah, exactly. I mean, yeah, exactly.
So like for endogenous retroviruses, for example, I have a paper, that I can pull up, but for sake of time, it says in that paper, it admits it comes from the conventional literature. If we as humans and mammals in general did not have endogenous retroviruses, actually this one's specifically focusing on a HERV, so human endogenous retrovirus, it says we and mammals would not be here right now, as in because it's functional and embryological development. So isn't that good design? I mean, we need embryological development. The chimpanzee needs embryological development. Gorilla. So is it really a surprise from a creationist perspective that we would share these types of DNA units with the biological? Isn't that just good design, Matthew?
Yeah, absolutely. I mean, and again, I mean, the sequences might be similar, but of course, they take part in embryological development. It's like a very basal process, and so of course we see these kinds of, I mean, what they mistakenly think are endogenous retroviruses when they have a function, when they have cellular functions. It's the evolutionary ideology itself is what is causing like a lot of misunderstanding in the scientific interpretation of all this data. But as to, and again, the evolutionists, they make these phylogenies, but as a bioinformatician, I mean, I've used these tools before. Many of these tools, they are based on producing evolutionary outcomes. Like, for example, there's the Blossom 62 matrix, and it's like they measure distances between two proteins. This matrix is used to find, if you have like a query sequence and you want to find like a similar sequence in the database, they use this matrix to find hits in the database. And so this Blossom 62 matrix, it just presupposes that it's built on like they use what the evolutionists, when they make this matrix, they use what they assume to be evolutionary relatives, and so they say that it's very likely that proline will be matched to a threonine or a serine, meaning that even in very well-known, widely used bioinformatics algorithms, you have these evolutionary presuppositions built in. And so again, like evolution in evolution comes out.
Right. Yes. And we discussed this in our last program, but I think it's such an important point because I've yet to see an evolutionist counter this. And we're focusing on the unconstrained sequences, hierarchies, DNA function, so on and so forth. Clearly, the junk DNA argument does not work for many reasons. We touched on a good one there with energy and resources for the cell. But if mutations are accumulating, they're accumulating quickly every generation. Even according to the evolutionary model, they'll try and say, well, a lot of them are strictly neutral or they get absorbed by junk regions. You've already demonstrated that's not the case. There is no such thing as junk DNA. But with Dan's challenge there, as creationists, you shouldn't have hierarchies and unconstrained sequences. Isn't it actually the other way around? Because if we've been evolving for hundreds of thousands to millions of years, I'm saying hundreds of thousands because they've got all these bottlenecks where they reset genetic diversity in a way. So if we're evolving for hundreds of thousands of years, accumulating mutations every single generation, if evolution's true in those neutral, unconstrained sequences, wouldn't that actually scramble those hierarchies where we shouldn't find? Because the evolutionists, they'll point out, well, humans and mice or humans and fruit flies, we share these genes. We have the homology there. Should that even be the case if deep time evolution were true? Because all that time, mutations accumulating, shouldn't they scramble those signatures, Matthew?
Yes. Well, I mean, That is something to consider. I mean, there's something called back mutations, and so back mutations they can also confuse the picture. Like they can confuse like the relationships, the alleged relationships that different species have with one another. For example, recently talked with Nathaniel Jeanson, and he studied I think mitochondrial DNA sequences, and he doesn't study the hypervariable regions because it's hypervariable. So there's so many accumulated back mutations in that part of the mitochondrial genome that it simply scrambles up the any kind of signal that he can read out of that sequence. And so yeah, so that also is a problem for evolution if we allow for them to have these unconstrained regions.
So our model, we have 4,500 years. We've been accumulating mutations since the flood and since creatures came off of the ark and diversified, adapted, and speciated in many cases based on the initial design diversity. 4,500 years isn't really going to scramble any kind of signal. The fact that we even have a general hierarchy and we have this level of homology, that's more consistent with biblical creation than it is for common descent, isn't it, Matthew?
Yes, because I mean, and I think I responded to one of Dan's videos on viruses. So we take viruses, and viruses have a very rapid mutation rate. And also consider that viruses are the most populous biological entities in the world. It's like, I think they're like 10 or like 100 times more virus particles than there are cellular organisms. Now, so they have a lot of particles, they have a very fast mutation rate. So we should be seeing viruses mutating into non-viruses. Especially since Dan Sterncoin and I, he believes that he believes in some sort of replication world. It's sort of like I think it's like a mythical world where you have these small replicators, and they're happily replicating, and they're replicating, and they're replicating. I think it's a myth because evolutionists really only induce this replicator world just so that they can have some sort of initial replicator to start the evolutionary theory. But so viruses, there are lots of virus particles, their mutation rate is going very fast, yet we see like virus meltdown. So this really demonstrates very clearly the nature of mutations through viruses, that viruses are not evolving into something, into non-viruses. They're not building up something new. Rather, viruses are undergoing mutational meltdown and their genomes are breaking apart. It's exactly the opposite of evolution, of what evolutionists want to prove. So viruses, I don't understand why Dan would be a virologist. I mean, he should know that viral mutations are just simply going too fast for viruses to turn into something really novel.
He's a virologist, and if he was looking at the data as objectively as possible, he'd be forced to conclude that common descent isn't true.
Yeah, right. I mean, I did bring this up in one of my videos, but he did not respond to it. So maybe it's another thing that we could ask Dan to do, to respond to if he is watching this video.
Well, I love that you put out several challenges just in these last two programs to the critics, so they have a lot of homework to do.
Yeah.
And I'm curious as to your thoughts, because the origin of retroviruses in general, from my understanding, retroviruses require a host to replicate. And so is there a chicken and egg problem for the evolutionists? Because if we were to ask them what came first, the retrovirus, well, wait a minute, the retrovirus needs a host to replicate, or the host. But if they say the host, well, then in a way, they're kind of admitting to the creation model, where I know a lot of creationists would say that some of these harmful retroviruses could have come about after the fall from pre-existing cells in host G. So could you speak to that?
Yeah. So where do viruses come from? You know, it is a very interesting question. And according to the creation worldview, that there was the fall, and so that's how harmful mutations could have accumulated. Viruses could have gotten out of control. They could have acquired they could become virulent. Although if we look at it, harmful viruses are really the great minority of all viruses. So viruses, could have played some sort of role before the fall, but they also could have broken out of cellular, out of cells. That's where they could have originated from. I also looked at they're called nucleocytoplasmic large DNA viruses in one of my videos, and so these are very interesting organisms. Some people think they are viruses. That's why they call them nucleocytoplasmic large DNA viruses. However, others think that these viruses, they're not really viruses, they're degraded bacteria. Yeah. I brought the example of the Cafeteria Rundbergensis virus, which has a genome of 1 million base pairs. So these NCLDVs, they don't really have like a specific name. They sort of like look like fuzzy, maybe like kooshball type particles. They're very, very extraordinary. I think they could easily be like a new domain of life, like eukaryotes, bacteria, archaea, and then you have like NCLDVs. They have a genome of 1 million base pairs. Some of their genes have cellular functions. It's like some of these genes in this particular Cafeteria Rundbergensis takes part in like sugar metabolism. So it's very likely that these NCLDVs are degraded. They are degraded bacteria, and of course, this again shows that left to themselves, organisms don't evolve into higher organisms. Rather, their genomes degrade, and so their phenotype also degrades along with it. So I think that these NCLDVs, they are very good examples of genomic degradation, not evolution. I did a separate study of trying to annotate the genomes of about like a dozen or so different NCLDV strains, and it's very interesting that up to like maybe like a quarter of their genes you cannot match with anything in the NCBI database. So these NCLDV organisms, they have a lot of orphan genes, not just one or two, but like a big, huge chunk, like 25%, 30% of all their genes in their genome. You can't really assign any function to these genes. They're novel open reading frames. They're novel genes, and so this is just yet another bit of evidence. I would have liked to talk about NCLDVs in my slides, but didn't have time doing the slides. But now we can talk about them. In fact, my first application of the gene content method was on these NCLDV organisms, finding different kinds of these NCLDVs. So they're very interesting. If anyone has the time, just look them up, Google them. They're very interesting, and they are proof for creation as opposed to evolution.
Well, it's another very helpful response, and I feel like some of the points there that you made will refute some of what the critics are saying in the chat. And so I'll bring this one up. And so what you were saying is it's the exception in terms of viruses that are harmful to begin with. I've heard it said, and I've read that there's more viruses in and on our bodies than there are bacteria or cells. And most of them are good and healthy and perhaps
most of them they are benevolent. They're like passenger viruses, so wow. they're not harmful. Okay, so one of the handful of people on the Ark must have been very sick if they had to carry all the viruses. That is, if we presuppose that they were sick when they got on board the Ark, and there were only 8 people in a very large area. The Ark was very huge; it was the size of an ocean liner. And I mean, granted that perhaps the 8 people, they could have gotten sick. Let's say Shem gets sick on the Ark. I mean, the Ark was very large. They could have had like a medical room or like a quarantine area where he could have possibly stayed until he recuperates.
Now, because the pre-flood world would have been so much different, the real estate was completely changed because of the flood. The environment was changed. I'm assuming mutation rates were faster, more deleterious mutations accumulated after the flood. And so as species are moving out and mutations are accumulated, environments are more harsh, could that be the perfect recipe for a lot of the harmful viruses to actually come about after the flood rather than before the flood?
Well, yeah, so I mean, you have these new living species, and these organisms, these species are splitting up, and so if if the organisms in a species, if they stick together, they're able to better maintain heterozygosity, meaning that their genetic variation is higher, and so with that, they're better able to adapt immunologically. Whereas if they split up into like smaller groups, um, it's like the founder effect. It's like they take those genes which are characteristic of that smaller group, and so, for example, there's a creationist hypothesis that says that Homo naledi was like a small group of humans, and that they went to southern Africa where they were discovered, and it's possible that they suffered from like a short stature, like a growth hormone defect. Neanderthals were also like a small groups of humans in Siberia. There's a cold environment. It's after the flood. You have the ice age coming on. It might have been freezing. They could have suffered since they were maybe like a small splinter group. They didn't have this heterozygosity present in their smaller population, and so they could have suffered more. They were more prone to illnesses like scurvy, gout. That's why they could have had these larger, more bent bone phenotypes.
Well, you mentioned some of the hominins there, Naledi, Neanderthals, and I think it's perfectly said that after the flood, especially after Babel, as people groups moved out, and some people groups maybe became isolated in less than ideal conditions and environments, and inbreeding took place, and you could have reductive evolution, as some call it, which would explain smaller body sizes, smaller brains. But I want to focus on Neanderthals and their genetics, because one of Dan's favorite arguments is, well, Neanderthals can't be descended from Adam and Eve because of their genetic diversity. They have too many mutations. The simplest way to explain it, and we could get technical on phylogenetics, but the simplest way to explain it is Neanderthals have too many more mutations than we do as modern Homo sapiens, that there's just no way we could account for all those mutations in the young Earth timescale.
Okay, so too many mutations. Well, I mean, we'd have to look as to where these what kind of mutations these are. I mean, we looked at last time. We looked at mitochondrial data, and Neanderthals, they cluster very well with modern humans.
Right.
I think I also did a study with mitochondrial DNA between chimpanzees and modern humans and Neanderthals and Denisovans, and so really the major difference was between chimpanzees and humans, and so and again I showed some evidence using a gene content method that when you look at like gene content. Neanderthals, they cluster nicely with modern humans, Neanderthals, and also Denisovans with modern humans, as opposed to ape species. So I think that despite Dan's contentions, and again, I mean, mutations can accumulate much faster than evolutionists are letting us onto. In fact, we can look at I think Dan also talks about the mitochondrial Eve, and mitochondrial Eve. There was a paper by Parsons et al. In 1997. Dan references articles from 1999. There was a 1997 article from Parsons et al. They studied the mutation rate. I think it was a mitochondrial mutation rate, and based on the differences between all humans all across different people groups all across the globe, they found that there's like relatively small number of mutations across all humans in the mitochondrial DNA, based on a separate study measuring the speed of these mutations based on the number of mutations, relatively small number of mutations across people groups all across the world, and the relatively fast mutation rate would seem to indicates that this mitochondrial Eve is 6,500 years old. And so even Parsons et al. In their paper, they say that based on this particular genome, mitogenome region, I think it was some sort of hypervariable region of the genome, so it's not coding region, that they did their math, looked at the number of mutations, looked at the mutation rate. If they did the math, they come out saying that this mitochondrial Eve is 6,500 years old, and that's a very biblical age for mitochondrial Eve. It's like if you look at the data neutrally in an unbiased manner, like Parsons, these are evolutionists in the journal of Science. They do this study, and lo and behold, they find that mitochondrial Eve has a biblical age. That's very good evidence for our side.
Well, it's incredible evidence, and the video that Dan most recently put out did not actually challenge what you said there. He missed the point, because some of these papers you mentioned, Parsons from 1997. Before that was Howell, which was another great study. There's been others like Santos, and then Howell did a review, I think was in 2003. But they're looking at, like you said, the control region, which is the D loop. Yeah. These are hypervariable regions where those mutations are largely neutral. So Dan's trying to say in his video, but once you add in purifying selection, now mitochondrial Eve has to go back 200,000 years. He completely missed the point that these studies are looking at the control region, which those mutations are neutral. Mm-hmm. Parsons, for example, looked at the control region, got 1 out of 33 for the substitution rate. Yeah. He said it's 20 times faster than phylogenetic rate. And if we were to go with that, the reasonable conclusion would be Eve 6,500 years ago. Well, lines up with the Bible. So adding purifying selection, Matthew, doesn't save the day, because these regions are not subject to purifying selection anyways. They're looking at regions where the mutations are neutral.
Yeah. And of course, again, if you look at these, at the mitochondrial DNA, again, it's a presupposition that these non-coding regions, they're also non-functional.
Right.
Again, it's just a presupposition. What if we do some research and we find some function for these regions? I just talked with Nathaniel Jensen. He says that when you look at the field of mitochondrial genetics and mutation rates, there are a lot of differing results. People are getting different results, and that he recently, I think he did a study of like Native American, not just Native American groups, but he had a book, an entire book on haplogroups in mitochondrial DNA. So it's not true what Dan Cardinal is saying, is that AIG does not do actual science. Answers in Genesis is doing science. Nathaniel Jensen is churning out these books. He is doing science, so that's a false claim by Dan. And what Nathaniel is saying is that based on his model fits very well like the histories of different people groups coming from like a recent ancestor 4,500 years ago. So Nathaniel Jensen's lyrical model, it does work very well. He's getting good results, whether he's looking at the hypervariable region or coding regions inside the mitochondrial DNA.
Well, which is essentially a confirmed prediction of what he was saying. And I've got his book behind me, Replacing Darwin. He made predictions on the history of civilization. Like you're saying, tracing the history of civilization should be possible if mitochondrial DNA and the Y chromosome go back just 4,500 to 6,000 years. Then he published Traced, which is another great book that I have behind me. And then most recently, which you mentioned, They Had Names, where he's doing exactly that. He's tracing the history of civilization, and it's matching up beautifully with what the Bible says. And it's based on the empirical mutation rate in the mitochondrial chromosome and the Y chromosome. I'd be curious as to your thoughts here, because this is one of my favorite phylogenetic trees that Dr. Rob Carter has cited many times, and you can find it in a paper there that he published with Dr. John Sanford, but also in one of his lectures here. And this tree, based on mutations and DNA differences, I've never seen somebody like Dan address what you've put forth with the pedigree studies of Howell, Parsons, Santos. There's another one in 2022, Connell et al. But that one, if I remember correctly, looked at the whole mitochondrial chromosome, and it came out with a really fast rate. It was like 0.5 per generation or something like that. But okay, in some of the coding regions, there might be some purifying selection. That's okay. We're looking, and you said something important there, the D loop, the hypervariable regions that are not subject to selection. That doesn't mean they're non-functional. It just means that their functions are not as essential. Obviously, if I get a mutation in something that affects my heart or
then that's
respiration, selection is going to work. But if it's something that's affecting something still important but not as important, or maybe DNA redundancy, it doesn't prove non-functionality. But here we've got overall low genetic diversity, Dr. Cserhati, where the mitochondrial DNA between any two people, separating them from Eve, the Eve consensus sequence, is only 20 to 30 differences. Max in some of the deeper lineages here, in some of these African tribes, are about 140, still trivial. And so the point is, based on the mutation rate, overall DNA diversity, the mutation rates that we get from these pedigree studies, this tree is only a couple hundred generations, which fits biblical time. And so my question to evolutionists is, how do you get 200,000 years out of the mitochondrial chromosome? I mean, where's the thousands and thousands of mutations, Dr. Cserhati, if evolution's true?
And exactly, and of course, Rob, he made this video, the three daughters of mitochondrial Eve, and we know that mitochondrial genome is maternally inherited. And so what Rob shows in that tree, I know that tree, and that basically there are three branches, like close to like the root. There are 3 points from which we have three major lineages, three major branches, and it's very interesting. Like, why exactly three major branches? And
that's incredible.
We know that Noah had three daughters-in-law. They made Noah's three sons, and so they passed on their three different mitochondrial mitogenomes to their descendants, and so those rapidly spread. Yeah. Okay.
We're just getting some positive feedback from the chat. People are really blessed by your work. And something that's always a hit is when us creationists, especially those like yourself with a PhD in biology, and you've done so much work on baraminology and created kinds, when we have brothers like you that are countering the critics, that are right in the battlefield and showing why the best that they have to offer simply doesn't work. On Neanderthal DNA, there's more mutations, or they nest differently from humans. I have at least one paper, and it talks about how some real families in the real world today, being subject to some like excess radiation, for example, or the various chemotherapy agents. I could pull it up. I'm just going from memory. It's in my slideshow somewhere, but it talks about how those families, because of the stressors that they're confronted with, they had a mutation rate that was 7 times faster. So they were passing on 7 times the normal amount of mutations. And so it makes me think you have which proves that you can't. Evolutionists can't argue. Well, this lineage has more mutations, therefore it must go back further than just 6,000 years.
Yeah.
Well, that's assuming that there's no conditions that can affect the mutation rate. And if you think about it, and perhaps you can speak to this, Neanderthals living after the flood, living in harsh conditions. Critics always say, oh, the flood would have produced a lot of radiation, heat, and energy. Sure. Okay. So this paper would tell me that if Neanderthals or other hominins like that were exposed to rocks containing excess uranium, they could be passing on 7 to 10 times more mutations to the next generation.
And that is a very good thought, because I mean, also, so this is one possibility, and also Neanderthals, they lived in the past, and they were isolated groups. So there's like the founder effect, and so all these three factors, they just add up to produce these larger genetic differences between Neanderthals and modern humans. And again, in my last time I showed the mitochondrial DNA, and so Neanderthals were seemingly a bit different, and somebody from the audience asked, like, isn't this proof of nested hierarchy? And it's not, because you have to take into consideration that this sequence comes from a Neanderthal bone like thousands of years ago. We're comparing it to modern human. Of course, there are going to be differences. So there are factors which cause the differences in the number of mutations. Yeah.
Amen. And I love that we have the real-time experimental data to back, because we're looking at historical science. Like you said, Neanderthals are in the past. Denisovans are in the past, Erectus. We got papers supporting, even on Dr. Carter's tree there, you can look at the HVR haplogroup, which is the most common in Europe, I think. And you see these differing size branches where some are going out here, some are a little shorter, but then they go connect to the same common ancestor. Well, that would be telling me that some people are picking up more mutations in the same amount of time than their cousins. Yeah. So how can evolutionists make assumptions based on a strict molecular clock? Well, they can't.
And also, I mean, so when you look at Neanderthals, we talked about their molecular genetics, but Neanderthals, they found fossils in Siberia, in like the Altai Mountains, they're like Denisovans. Denisovans are like similar people group. In Spain, Northern Africa, Morocco, and they have found like Neanderthals, like these mixed fossils, which have Neanderthal and also traits from modern humans. So it's not purely Neanderthal, not purely modern human, but it's like a mixed modern human and Neanderthal. They're mixed together. One thing, and I think that that they did find an example in, I think it's Vindhya cave in Croatia. It was a Neanderthal fossil wearing armor, meaning that he was from the Middle Ages. And so that definitely proves that Neanderthals were humans just like us. In fact, if we talk about Neanderthals, it's a very good topic to talk about. In 2015, evolutionist authors came out with a book called Rediscovering the Neanderthals, whereby they said that we really have to rethink everything we know about Neanderthals, because modern science has simply discovered so many things about them. That Neanderthals, they used aspirin, they made tools, they made tool-making tools, they had religious ceremonies, they dove for shellfish, they dove underwater, they had hunting patterns. So they behaved and they exhibited behaviors which would show that they really truly were very much, I mean, they are humans just like us.
In my deep dive into the hominin fossil record, I listened to a lot of lectures from paleoanthropologists that are non-creationists. And there's one relatively recent lecture where the speaker said Neanderthals, and remember, this is an evolutionist, he said Neanderthals are us. Neanderthals are human in every way, just adapted to the cold ecoglacial environment.
Yeah. And so, if you think about it, we shouldn't judge fossil hominins based on modern humans. Right. It's like the arrogance of modernity. We shouldn't call the modern humans like humankind. We shouldn't say that we are humankind. Rather, the human species, we should look at it as a continuum moving from the past into the present. So African Americans are human, Orientals are human, Hispanics are human, Caucasians are human, Neanderthals are human, Nadius is human. It's just that the variation within humankind is greater and maybe something that we might not even expect, but still it is human. We are human. We shouldn't judge like past humans based on what we observe today. Rather, we should see humankind as a totality of all individuals, from Adam to Noah to Abraham to David to Christ, and then to us in modern times.
Well, it's a very interesting point, and it brings a question to mind. If today we're looking at modern humans, extant humans, we have a specific phenotype generally, but then you look at hominins in the past and you have a slightly different phenotype in Neanderthals, in Erectus, Heidelbergensis, and you don't really find the modern human phenotype in the so-called fossil record, or maybe you do. We'll see what your thoughts are on that. Therefore, in your opinion, what do you think Noah and his family, and then going back into the pre-flood world, do you think the pre-flood world phenotype for humans was more similar to us as modern humans, or more similar to something like Heidelbergensis?
So what were the pre-flood humans like? What was Adam and Eve? What were Adam and Eve like? They would have had like a more robust genome because again, like Noah and his family, they underwent a bottleneck effect. And it's interesting that even secular evolutionists will claim that recently the human genome pool underwent a bottleneck.
Right.
We know that that's the flood. We can put a name on it. They're trying to figure it out. They're trying to put an evolutionary explanation to it. So what did they look like? I think that pre-flood humans, the variability would have been greater than modern humans. Again, although today there are 10 billion of us, so 4,500 years versus 1,500 years, 4,500 years of creating 10 billion individuals, that also accounts for quite a bit of variability. I mean, we are still humans. I think it's interesting that if you look at, let's say, black people and white people, there are both white people and black people. There are some people who are tall or short, fat or skinny, smart and dumb, not so smart. It's just that really the only difference between these major people groups, skin colors, is really the color in skin. They did a study of like albinos in these African tribes, and this one African boy, he was an albino. He had white colored skin, and so you look at him, he sort of resembles like a Caucasian kid, like a white kid. You just don't realize that he's African. It's just that he's albino. He doesn't have this brown skin color, and so skin color is really only like a very, very infinitesimally small difference between people groups. And so overall, humans, we all are quite similar. I mean, again, there's only like 10 million positions in the human genome. That's like 10 out of that's like 0.3% of the whole entire human genome, which accounts for all differences between humans.
Wow. Yeah, I think I've heard it said that just even within us, we have 3 million spots that differ. Common alleles it's a 10 million. And okay, so then there's a bottleneck on the flood. Let's say even half was lost. You can throw 15 to 20 million into Noah and his family. Same thing roughly for Adam, and that would explain the diversity we see today. You have some rare alleles as well, since spreading out at Babel, blue eyes. I know that's isolated to certain people groups, and sickle cell anemia. And so you're saying from the Cretaceous perspective, with created heterozygosity 6,000 years ago, and then the bottleneck, and then Babel, it's pretty easy for us to explain.
Yeah, it's not difficult. I mean, look at what the evolutionists have to do. They have to explain how all of life, all life forms, all 100 million species, how they originate from an original single cell. I mean,
wow.
I don't envy them.
No, I don't. And that's why I said after your presentation, I said, I wouldn't want to be an evolutionist who believes that life came from non-living chemicals in some nice warm pond billions of years ago, and then single-celled to multi-celled up to us as humans and whales. I mean, that's a difficult task. For us, it's from humans at creation, humans at the flood, to all the humans today. And I'm curious, so there's something out there which I believe you've answered sufficiently, but evolutionists still put forth what's called the phylogeny challenge, how to answer the question of ancestry, which you've shown us how to draw lines. You have drawn some lines as well. But when we look at creatures like canids and felids, so cats and dogs, we have a lot of variety, let's say in dogs, coyotes, wolves, dingoes, domestic dogs. But then when you bring in some creatures that maybe they don't look quite as similar as what I just named, but they still kind of look dog-like, how do we draw a barrier between? Is there enough differences and discontinuities between families like felids and dogs for us to say for sure these are different kinds?
Well, you see, I think that if you look at morphology, that's the big drawback of morphological methods. You see that even the same species, even the male and the female of the same species can be radically different. There's some marine creatures where the male individual is so reduced that it only serves sort of like as an appendage to produce sperm cells to inseminate the female. Wow. That's how drastically that particular species is reduced. So with morphology, we see that there's a lot of variation, and there's a lot of noise. And so that's why, as a molecular biologist, I would say that it's better to look at the molecular data, because again, the genes, they determine, the genotype determines the phenotype. The genes are simply more basal than the phenotype. And so that's why we can do a lot with mitochondrial sequence similarity, or we can do whole genome sequence analysis, or like gene content method. And then we can do clustering analysis. We can also like calculate p-values. I also mentioned that we can look at the Hopkins clustering statistic. What this statistic measure does is it takes a similarity matrix, and it will tell you whether there are real clusters in your data. And so it'll give you like a number between 0 and 1. The higher this number means that there are real clusters in your data. Now, evolutionists, they approach things from like a phylogenetic standpoint. That's their do-all and all. That's their thing. I mean, you can approach data analysis with from a different standpoint. Why not do it using clustering methods? You have like matrices, you run your algorithm, you get clusters. These clusters, they correspond to hypothetical kinds. That's a different way of approaching things. Some people have evolutionary presuppositions. I try to find clusters, and so I use k-means clustering or other types of clustering methods. It's just a different approach.
So if you have 3 different groups of creatures, like group A, B, C, and then you do this analysis, if they cluster with enough discontinuity between the 3, is that how you conclude that this is a different kind than?
Yeah. So like I also showed the elbow plot. What the elbow plot is, the way the k-means clustering algorithm does is, let's say you have like a 2-dimensional plane, and you have all these dots. And then let's say you draw clusters around these dots. and then you compare the distances within the clusters compared to between the clusters.
Okay.
And then you divide, I think it's a total within the sum of similarities. And so if you achieve an optimal number of clusters, then the total within sum of I think differences, or the sum of the differences will become very, very minimal, because previously, in a previous step, if you have a cluster with very distant species, you're going to get distances which are very big. And so that's why at smaller cluster numbers, you'll still have like a high total within sum of, I forget what TWS means, but you'll have like a very high value for the within cluster distances. As you achieve a better optimal clustering, these within cluster distances will drop dramatically. And it's at one certain point you're going to have like an elbow. That's why it's called an elbow plot. And where the elbow is, that's your optimal number of clusters. So this means that we have clustering tools whereby we can tell you what the optimal number of clusters are within a data set. It means that we'll be able to give you a list of kinds. What's also interesting is that there's also another similar parameter called the silhouette width. It also I sort of forget how it goes, but it's also based on clustering the species differently and finding like an optimal clustering and optimal partitioning of the species. And this time, instead of within cluster distances, you're measuring the so-called silhouette width, which is, I think it's like the width of these clusters. Instead of distances, you're calculating the width of a cluster. And so you can also get like an optimal number of clusters using this silhouette width parameter. There's another algorithm called the Kalinsky algorithm, and it'll measure like silhouette width for a number of cluster numbers for you. And where this Kalinsky parameter is optimal, it'll tell you that that's the optimal number of clusters in your data set. So it'll tell you what is. So basically, biologists, we can use this Kalinsky criterion. We can use like half a dozen tools to find the optimal number of groups in your data set.
Wow, it's such an awesome skill. Yeah. And I have several of your papers in front of me, which you've done clearly a lot of work on baraminology. And I'm curious if you ever find overlap in your clustering and answering the question of kinds that you weren't really expecting, where you thought, oh, this kind of overlap or the way that these creatures clustered wasn't what I was expecting, in the sense that I didn't think that this would be related to this.
Yes. So again, biology is messy, and of course, I mean, what kind of researcher finds perfect results all the time?
Right.
It tells me that there's an added level of analysis or interpretation that you have to go through to make all things snap together. What I have found is that, for example, in chloroplast genome is larger than the mitochondrial genome. And sometimes what I find is that, let's say I'm looking at a genus of plants. I find that in my heat map, let's say if you have a heat map, there are chloroplasts, let's say there's members of this particular genus in this corner and also in the upper left corner. Why is this genus, why is it separating for me? And several times I found that an inversion has happened inside the chloroplast genome, which may affect even up to a third of the chloroplast genome. Like a third of the chloroplast genome simply gets inverted. And so what happens is, if you're trying to line, let's say you're looking at, let's say some sort of myrtle tree, and two species from the same genome, you try to align their chloroplast genome, but in the middle, if you have this inversion, that middle third portion of their genome is going to drastically reduce the sequence similarity because the sequence is the other way around. The sequence is simply messed up, and so that drops the overall sequence similarity between these two species in the same genome because of this inversion. And so I found that when I straighten out this inversion, these two species in the same genome, they now come together. Because overall, that inversion mutation that is really only one single mutational event, and
Right.
that's what causes that's what sort of screws up the heat map. But it's only one single mutational event.
And this is the more effective way, you were saying, of answering the question of ancestry by looking at genetics, genes, its traits that's passed on, sperm and egg.
Yeah.
This is the direct way to answer the question of ancestry, universal or separate ancestry, like the mitochondrial DNA is, you focus on that DNA compartment. Over looking at like just more, because you were pointing out all the problems with more, even just a female and a male species of the same animal could differ drastically in terms of morphology. So it's more important to look at genetics than morphology, fossil data, anatomy.
Yeah, because simply the genotype is more basal. The genotype determines the phenotype. And, and of course, I mean, I'll be candid. I mean, what kind of researcher gets perfect results all the time? There are species which sometimes makes me wonder. But then again, I mean, when we get these cases, I mean, there are these cases. Can we always chalk it up to evolutionary continuity? That's the evolutionist basic default mode of thought, that if there's if there's no clear cut, then obviously they must be related. But in many other cases, there are species which are, or like clades, which separate very well from other clades in these phylogenetic trees. They're separated by these very long branches. I showed a case of these. I think it was this mouse family that it had like a small species number, but it was connected to all the other mouses by this very long, separate, isolated branch. And so these evolutionists would have us believe that in these branches leading up to these separate families, there's like a rapid burst of accumulated mutations. How does it happen? What's the reason for this? Sort of murky. I mean, it's I mean, so it's sort of like you're saying that if I want to build up this new gene sequence, I'm just going to randomly change a bunch of bases, or let's say amino acids, and then if I play it right, I'm going to just by randomly making all these changes, I'll get this new protein sequence. It's sort of like, again, like winning the lottery, not just with like 5 numbers, but with like 200 or 300 numbers, and you get all 300 numbers right.
What about, because I also know that you've done some baromenological work on some of these interesting, I've got one of your papers in front of me here on Archaeopteryx, I believe. And so what are your findings on some of these creatures that evolutionists say are transitional, or we might say are mosaic? When you look at well, I guess with Archaeopteryx, you wouldn't have the genetics. So how are you trying to figure that out to see if it's what it's related to, where that breakoff point is?
Well, I would say that Archaeopteryx is a bird. I think it's too bird-like. I mean, it may have like a long tail. Some of these bird species might have like sharp teeth. Again, I mean, it may be that birds lost this genetic information for sharp teeth or for tails.
Right.
But again, like overall structurally, its limbs and its tails would make us believe that it's better suited for flight. Reptiles, their legs are a lot larger, and simply like the balance between reptiles and birds would make it like impossible for a reptile to undergo certain transitions to turn it into a flying animal like a bird. But again, I mean, just don't discount that that Archaeopteryx doesn't have any DNA. Again, it's just since the evolutionists presuppose that these are all ancient fossils, they're millions of years old, they can't even possibly have any DNA in them.
Good point.
I'd like to challenge the evolutionary community. Just try to find mitochondrial DNA in Archaeopteryx or Homo naledi or all these all sorts of fossil organisms. Maybe there is some sort of remnant, even if it's like from a single gene, like cytochrome oxidase 1. Maybe there is DNA there. I mean, they allegedly have found DNA sequences from dinosaurs. So that would be interesting. Let's not stop scientific thought that, you know, it's millions of years old. There can't possibly be any DNA there. End of story. You don't need to look any further.
Well, I think that's a great point, and that comes down to making testable and falsifiable predictions, the gold standard of science. Whereas at Cretaceous, we believe that the so-called geologic column, all the rocks, and you could debate flood boundaries and so on and so forth, but we would believe all of this is representative in terms of biology and the Earth 6,000 years. So we could make a prediction, especially after the flood, some of these so-called hominins like Naledi, some of these Australopithecines, we could predict, okay, if we actually go looking and don't assume billions of years, perhaps we can get some DNA, some mitochondrial DNA. And ahead of time, maybe we can make some predictions as to what would be related to what. Like Australopithecines would, you know, we'd find enough discontinuity in their genetics to separate them from humans. Yeah. Or with Naledi, we'd find enough there to suggest they are humans. We can make those predictions ahead of time, couldn't we?
Yeah, and exactly. I did do a paper, and they actually found mitochondrial DNA for Homo heidelbergensis, and I was just overjoyed. Okay, I immediately downloaded it, then aligned it with Denisovan, Neanderthal, modern human, and a lot of primate species. And the prior creationist prediction based on morphology was that heidelbergensis is like an erectus, and so it should be human. And so, lo and behold, that's what I found. I found that sequence-wise, Homo heidelbergensis clusters together with the human. So both morphologically and also genetically, from molecular evidence, Homo heidelbergensis is human.
Wow. And that's the joys of science. That's one of the amazing part of being at Cretaceous is science is always advancing. We're getting more data, and it always confirms biblical creation. Eric is wondering what evidence would disprove.
Okay, so here's my answer to Eric. Okay, now, in a sense, Dan Cserhati and I, we are both Bermanologists. Now, you take the big evolutionary tree. Imagine that you can make this big evolutionary tree, and so that consists, as we know, of 100 million species. Now, evolution strictly says that we have one single tree, we cannot deviate from it. What I'm saying is that if we find anything that disproves relationships between, let's say, vertebrates and invertebrates, the evolutionary tree is going to fall apart into two trees. Now we have an orchard. That's a creationist orchard, as opposed to the single evolutionary tree where everybody is related to everybody. So Eric, imagine that you've got imagine graph theory. You've got all these dots in the two-dimensional plane, and you're drawing a line between two species, two dots, which are related. And so in evolution, you have to create a single tree. Creation theory says that we have a forest. Now, according to graph theory, how many possible forests can we have? We can have anywhere between 2 and 100 million forests. 100 million is when every single species is its own tree. The only single scenario where evolution would win is if everybody is connected. And so I would say that the overall probability, overall likelihood is that the probability that the evolution is true is 1 divided by 100 million. So that's my answer. Evolution is simply so improbable. I mean,
that's a really low chance that we're related to grass then.
I mean, what evidence would disprove evolution? I'd like to ask Eric the same, the counter question.
Right.
I mean,
Well, and we've shown so many, and you've shown so many major discontinuities and differences. In the last program, we talked about even just the Y chromosomes between humans and the great apes, massive difference, less than 30% between humans and chimps.
Yeah.
And so the evolutionary community has.
Yeah. It's another big major genomic disjunction.
Perhaps I want to respect the time of the program. Time always flies by with you, Dr. Matthew Cserhati. So we're going to wind down here, I guess, with one or two questions. Another truly informative. I feel like I could just sit back and just ask you questions for 10 hours straight, because I always learn from you and you're always very informative. Mm-hmm. So as you were presenting and refuting Dan on the viruses point, there was some back and forth in the live chat. So I did catch this. And Chris is trying to say, well, Dan, this is referring to Dan, is saying that viruses show what separate ancestry would look like in polyphyletic genomes.
Yeah.
We do. So he's saying we don't see that pattern in cellular life. We see patterns that would indicate common ancestry.
Well, what we see in the evolutionary tree, I mean, actually, has any evolutionist produced an evolutionary tree which contains all 100 million species? If you can show me that would at least model the evolutionary tree. I mean, and again, we don't have the ancient DNA. We just simply don't have the stuff to produce this big major evolutionary tree. And like I said, that and evolutionists themselves know very well that between clades there are these big cracks. Like the NCLDVs, like 25% of their genes are orphan genes, which really very much separates them from the rest of life. So whether you want to admit it or not, there simply are a lot of disjunctions in the evolutionary tree. So much so that, in my mind, the evolutionary tree is falling apart into a lot of individual trees, which then turns into the creationist orchard.
Which is every time they invoke convergent evolution, they're admitting to, once again, a lack of uniqueness in the universal tree of life. So polyphyletic. So is Dan trying to say that because all viruses don't come together as one group or one evolutionary tree, that confirms evolution and contradicts creation?
So what a back and forth debate was on YouTube was that we know that viruses are polyphyletic. They do have like independent, separate origins. So that's not a point of contention, that viruses have separate origins, which I showed. He's showing. I showed with my gene content method that viruses are polyphyletic. They have separate origins. So that is evidence for separate kinds. What Dan is not understanding is that I used the exact same algorithm, this gene content method, which I co-discovered with evolutionists. I just found that this Dutch evolutionist group, they came up with the same method. So it's not my method only. Using this method on data from cellular life, I found patterns. I found patterns of polyphyly of statistically significantly different clusters of cellular life forms. So if we're consistent, if we accept that the gene content method finds kinds in viruses, I've used it in NCLDVs in primates, and I think in like other groups, we also find groups in cellular life forms. So I'm not understanding like what is Dan's problem. I'm consistently using.
You're being consistent, right?
The same method in different data sets, and I'm finding kinds in viruses and also in cellular life forms.
Wow. Okay. So let's reiterate that challenge then to people like Dr. Dan and Aaron Ra and all these militant, prominent atheists. And correct me if I'm wrong, the same data sets, the same methodology where you're finding separate kinds, separate ancestry and viruses. You're using that on cellular life, and you're finding separate kinds there as well.
Separate kinds. I mean,
Multiple trees.
if evolution is true, if I'm using the gene content method, I should have like a very low Hopkins clustering statistical value.
Right.
It's very high. I find real clusters. That's what Hopkins is telling me.
Wow.
Yeah, I mean that these are simply the results.
Real clusters suggesting separate ancestries.
Suggesting separate ancestry. These clusters, which I correspond to kinds.
Wow, that's amazing. And right there's a major and that's why the probability or the chance of evolution being true, common descent, is just so incredibly low.
Yeah. So I mean, if evolution is true, I mean, we should have like a big smear of like all the species they're like all interrelated with one another.
Right.
We shouldn't be finding statistically significantly differing.
Are you saying we should be finding a lot of continuity all the way through?
Like continuity within all the species in the data set. But as I detailed, the way clustering works is that the algorithm, it clusters species, and then we compare the similarity values from between species in a kind and between species in that particular kind and all other species in the data set. So we get these similarity values, and then we have two sets of similarity values. We can run a student's t-test on these two sets of sequence similarity values, and the student's t-test will give you a p-value. If the p-value is less than 5%, it means that there's a significant difference. The p-value, it'll unite continuity within a kind and discontinuity between the kind and non-kind into one single number. And so I think that you've got morphological baraminology, but if you add statistics like Hopkins clustering value, p-values, Kalinsky index, silhouette width, elbow plots, and if you look at the molecular data, it'll give you a more clear, crisper picture of how there really truly are kinds out there in nature based on these things.
And when you, as a final question, when you do that with species that we as creationists assume are related and go back to the same archetype, like let's take felids, lions, tigers, jaguars, cheetahs, does that correlate nicely with them all being related?
Yeah. So cats, dogs, horses, these are well-known kinds. And I mean, I've analyzed these groups before, and I have found that cats nicely form a kind. They're separate from dogs, separate from horses, and so we're just really validating that. I mean, dogs, cats, horses, they're visually different, and so we also find that their mitochondrial DNA is also statistically significantly different from one another.
Wow! Praise the Lord, Dr. Matthew Cserhati. I love your work. Again, another excellent presentation. You gave us more rebuttals to challenges and arguments from people like Dr. Dan Stern-Cardinal, Dr. Chris Thompson. In the previous program, you countered very thoroughly arguments from Gutsick Gibbon. And so the ball is in their court. Let's see if they can refute all of this overwhelming evidence supporting separate ancestry. Matthew, I always appreciate your time. I want to hand it to you for some final words, final thoughts, anything you'd like to leave myself with and our listening audience, brother.
Yeah. Well, anyway, so Donny, thank you once again for letting me be on your show. And so I just want to say that whenever I look at the data, I mean, I'm a bioinformatician. Whenever I get to analyze data, I'm really enthused. I can just look at the data all day long, trying to like analyze it in more and more depth. I'm trying to think God's thoughts after him. And I think that really we have so much data out there in bioinformatics, lots of mitochondrial DNA. I mean, there's so much to analyze. I am not intimidated by the data. I want to get my hands on the data. I want the evolutionists with all the research money and the grant money to go out there, find mitochondrial DNA in these fossils. They should go out there with all their money and their influence and get that mitochondrial DNA. I want to get my hands on it. to see like what the data really shows. And I mean, I've been finding lots of good results, which shows kinds in mitochondrial DNA, genome analysis. I'm really enthused, and so for the Christian audience, I would like to say that do not be intimidated, do not be discouraged. With more and more scientific evidence, we are finding functions for new genes, for all these unknown sequences. We're seeing clear differences between different clades, at least on the apobirmidic level. And so, I think that if you keep an open mind, if you look at the data objectively, fairly, you will see that the data really supports the creation viewpoint and not evolution. And so just trust in God. His word is truth. It is true from the beginning to the end, from Genesis to Revelation, and also in between. God is truth. Jesus Christ is the truth, the way, and the life. So we can trust in Christ as our Lord, our Savior, and our Creator. And so that's what I have to say for all of you in closing. So thank you once again, and thanks be to God. All glory be to Him.
Praise the Lord. Amen. What a great way to end another excellent, informative, and comprehensive program with the gospel and an encouragement to the lost to trust in Christ, because the Bible is true. The Bible says that we shouldn't put our faith in the wisdom of man, but in the power of God, because we see the fruit of the wisdom of man, which says that we're all related as humans to grass and whales, and life came from non-living chemicals. You've shown very thoroughly now in just these two programs, roughly 5.5 hours of material and content, overturning common descent, showing separate ancestry, answering the critics, answering the challenges, but also issuing many of your own challenges to the evolutionary community. So I want to see the evolutionary community, the apologists of common descent, to do their homework, because we've clearly done our homework. You've done the work, Dr. Cserhati. And wow, this has been just a great program. So I want to encourage those in the audience, share this around on YouTube, share it around on social media, please. Doki, my faithful mod brother, has been posting Dr. Cserhati's YouTube channel. Matthew, you are a brave, courageous warrior. I love seeing whenever you put a new video answering one of these critics, taking them on head on right in the battlefield. So go give Dr. Cserhati a subscription. He's got great videos. Check the description box for a link also to where you can read all of his articles and his technical papers, and check out for yourself all the work that he's doing in terms of these baraminological analyses. So wow, what a great program. Matthew, I appreciate your time. I look forward to hanging out with you again at some point in the future. I could ask you questions all day. To the audience, thank you for the support, the engagement. To the critics, thank you for the feedback, the challenges. Standing for Truth is signing off. God bless all.